In this video, we'll go over the answers for Wednesday, April 8th. And what we're talking about today is a continuation of the cell membrane. And so I have some slides here from our video lectures, and I'll just go over them because they're important to know to really understand what we're talking about today. First is the experimental technique known as FRAP, or fluorescence recovery after photobleaching. And the idea of FRAP is that you will take a protein and label it with GFP. If remember, GFP is a protein that natively has fluorescence when you look at it under a specific microscope. So what you can do is you can modify the DNA sequence of a protein you're interested in and have GFP be attached to that protein. And well, in this image, that protein is just all over the cell. Then what you can do is you shoot a part of the cell with a laser. And when you hit GFP with a specific laser, that GFP will stop fluorescing forever. So we get this white circle. Now, the recovery part of FRAP is that you will see after time, the white dots will move away and new green dots will move in because proteins can normally diffuse without a problem. So with this, you can measure diffusion rates of any protein. What this can also be used for is that if your protein has no movement, that is, it's fixed somewhere in the cell and never moves, this white circle will never change. There will be no new green particles to fill in this white circle because all the proteins will be immobile. And what we found when we do this for proteins in the lipid, or the membrane bilayer that is that most of the proteins are somewhat mobile. They can move around in the lipid bilayer. And we see that when we did this experiment. So we label a bunch of mice proteins in the lipid bilayer green, human proteins red, have them in separate cells, use a virus to combine these cells. And what you find out is that the green and red mix, showing that these proteins are free to move. And from that, we have come up with the fluid mosaic model of the membrane. Fluid because stuff can move around in the membrane. Mosaic because it's made out of a lot of parts. So we have our phospholipids, we have our cholesterol, we have integral membrane proteins, that is proteins that go all the way through the membrane and cannot survive without being in the membrane. We have lipid link proteins, which are attached to a lipid, that's how they say with the protein. Peripheral proteins, which are like at the head groups and can come off and come off, or come on and come off rather. Oligosaccharides to make glycoproteins, or we can put a saccharide on a lipid to send specific signals. Now, there is one other structural feature of the membrane we haven't talked about before, and that's spectrin. You can think of spectrin as the skeleton for the membrane. Erythrocytes are 75% spectrin on the membrane for proteins, and that gives it this donut shape. So spectrin is really the skeleton of the membrane. And spectrin, as you can see here, is attached to this protein called ankyrin. So ankyrin is the anchor for spectrin. And this is attached to an ion channel. You can kind of think of these as the stakes that you tie spectrin around, or spectrin associates with, to keep spectrin with the membrane, and then spectrin can go off like little fences. And different proteins can associate with spectrin. Here we have actin and tropomyosin interacting with our spectrin. If you remember, actin's in the muscle. We talked about that there, but actin's actually all throughout the cell, not just in muscles. It is the cytoskeleton of cells. And what we learn when we look at spectrin and GFP is that some proteins have different amounts of mobility in the membrane. So, for example, protein A is associated with spectrin, so it will never move. So if we do our FRAP experiment, we'll never see that white circle fill in because A is immobile. B has some movement, but it's stuck. See, it's stuck in this like little pen. So B can move a little bit in the membrane, but not all that much. while C has more or less free range of motion in that it can move around. And this is called the gates and fence model of the membrane. And what we see a lot when we look at proteins is that they'll be stuck in little areas and then eventually jump over to a new area. So, for example, C might be stuck right here, but will be eventually able to pass through the spectrum and go to this area and continue on in the membrane. So we see that quite a lot when we do our GFP experiments, and that type of behavior is thought to do because of spectrum. All right, so the first question gets at our ability to understand the fluid mosaic model. So which one's false? So A, if we see lateral movement, that means it's moving left or right in the membrane, even if the cell is anchored to a slide. So the cell can't move, but we still see movement in the membrane. Yeah, that's fluid mosaic. Transverse diffusion. So that's our false one, because transverse diffusion means I go from the cytoplasmic side to the extracellular side of the membrane. So that's not fluid. That's not really moving. That's just moving from one side of the membrane to another. Fluids, when we talk about fluid mosaic, that's more like moving left to right in the membrane. You're kind of traveling along the cell. Transverse is just you're flipping sides. So that's false. Membrane fusion, that's the experiment we looked at. Lateral diffusion. So lateral diffusion is what we're talking about, movement left to right. Why are some membrane proteins anchored and others free to diffuse? And so if you're anchored, whatever you do inside the cell or in the membrane, you have to do it in a specific spot or associate with specific proteins. So it would be a lot more convenient for the cell and a lot more efficient if that protein's always stuck there. While something that can diffuse, you can still do your job without having to be in a specific part of the membrane. So there's no reason to spend energy to hold you in one part. So you're free to move along. So that could be the functional reasons for these different types of movement we see in the membrane. Now, when we look at membranes, we'll see that each side of the cytoplasm has different phospholipids. And the enzymes that do that in humans, eukaryotes, are in the ER, the endoplasmic reticulum. Prokaryotes do that in the plasma membrane. Now, everything's made inside the cell, yet we have different phospholipids on the outside. So how do we do that? Remember, we talked about in the last lecture, lipid head groups are charged, so it's energetically unfavorable to move them from one side of the membrane to the other. For example, the cytoplasmic side to the extracellular side. So to help with that, we have a protein called flippase. Flippase is a protein that specifically just changes which side of the membrane you're on. So it will come and grab you if you're on the cytoplasmic side and flip you to the extracellular side. This is facilitated diffusion only, so it will only do it without spending ATP. So if we look at phosphatidylethylamine, most of it's on the cytoplasmic side. Some of it's on the outer side. So flippase could flip phosphatidylethylamine energetically, like for free, because of concentration. There's so much little on the outer leaflet, it's energetically free. We have phospholipid translocase, so that will transport specific phospholipids using ATP. That's active diffusion. And so sphingomyelin, a lot of it's on the active. You might have a translocase for that. Now what you can see is that phosphatidylserine's all on the inside, which means if it's ever found on the outside, that's a signal for something. Because normally it's not there. So if it's there, we can use that to say, hey, I'm being shown on the outside now. That is a code to the rest of your cells. So phosphatidylserine could be a code for blood clotting. It's also a code for cell death, for macrophages to come and destroy that cell if there's a lot of phosphatidylserine showing on the outer leaf. All right, so this question is getting at your understanding of FRAP. So could we use FRAP for concentration? No, you're just going to see a bunch of light. I mean, you could do relative concentrations, like I see more light with this protein, less light with this other protein. That's going to be hard to do. You can do it though, so, but it's not really useful for that, especially since you're shooting it with a laser. That's actually FRAP, and what I was describing is just more fluorescence. Diffusion of protein cell, yep. Like I said when I was talking about FRAP, speed of flipase, no. FRAP is used for proteins, not for lipids. Also, you the cell's 3D, but with FRAP, you're kind of only seeing 2D because you're looking right down on the cell. So you can't really tell what side of the membrane it's on because the cell looks like a 2D image to you. Lateral diffusion of a lipid in a cell. I have yes there. I mean, technically, if you attach your GFP to a lipid, you could. That's hard to do. GFP is mainly used for protein. So I'm going to say, now that I'm looking at it and thinking about it, probably not. Theoretically, yes, you could, though. All right, so talking about more of the plasma membrane, there are specific organizations of the membrane that are called lipid rafts, and they're like subdomains. They're little domains of the membrane. And they're basically just lipids and proteins that are associated together. And they might have different functions, and these functions are due to different amounts of lipids and proteins in there. And you'll see these lipid rafts anytime you look at a membrane. So there's a lot of glycosphingolipids and cholesterol, very organized, and certain proteins can associate with them. Also, some diseases use these lipid rafts to get into your cells. So the flu, measles, Ebola, or HIV, before they enter your cell, they'll first localize to the lipid raft and then go inside your cell. So for the shape of erythrocytes is due by what? So this is what we talked about earlier, just seeing if you're paying attention. That is spectrin. So I have question 4 again. So yeah, misnumbered here. This is looking at, do you know what side goes to, for the lipids that we find, what side are they normally found on? And here in the top, we have inner. On the bottom, we have outer. Your big key here, phosphatidylserine is only found on the inner leaflet, only towards the cytoplasm side. All right, the next thing we're going to talk about is how we can move proteins across a bilayer. And here we're talking about the layer of the endoplasmic reticulum. Because proteins need to be in the endoplasmic reticulum, but it's not easy to transport a whole protein. So what is the strategy for that? And so the way that we can transport proteins is when they're being grown from the ribosome, on the N-terminus, there's going to be a specific signal. And here are just some signals we have found for a bunch of different proteins. So you can see it's very hydrophobic, this signal. When this signal is being created, there's a protein called SRP that will come and bind this peptide. Once bound to this peptide, SRP, which had GDP on it originally, will get GTP, and it will escort this protein in the ribosome to the SRP receptor in the membrane. So when SRP binds, it stops the protein from being made, goes and finds a receptor, and this protein is guided into the translocon. The translocon is a channel that allows proteins to be to kind of travel through it, so they can go to the other side of, in this case, the ER, the rough ER. So the SRP goes to the receptor, they bind, the new protein's going through the translocon, the receptor and the SRP both lose GTP, and so the SRP falls off and the protein is starting to be made again. When the protein goes through the translocon, a lot of times what happens is that the signal peptide is cut off. There's usually a little protein right by the translocon that cuts off the signal peptide, and it will keep growing. And if it's a membrane protein, part of that protein will be embedded into the membrane. So there's another protein by the translocon that can help embed the whatever needs to be into the membrane. Not all proteins that go through the translocon have to be in the membrane, though. I'm just showing the one example that happens. Some proteins can just float off into the ER. But once our proteins are made, ribosome dislocates, and that's how you get proteins across membranes. All right, so this question really gets at your understanding of, do you know what I just said? So what happens if in your signal peptide you have a cut sequence, or you have a mutation that cuts between two leucines? So the signal peptides only cut after it goes to the translocon. So getting it to the translocon should be fine. That should be no problem. But what could happen potentially is if your translocon has two leucines in it, let's say right in the middle, then it's going to be cut right in the middle, while normally it should be cut at the end of this red sequence. But now you're cutting in the middle of the red sequence. And what that could mean is that your protein will now misfold because you have an extra amino acids that are there that are not supposed to be there. So that could cause misfolding, or it could cause nothing to happen. It would be on a case-by-case basis. So question 6, fill in the blank. So the translocon, which is in the endoplasmic reticulum, is a transmembrane pore. So you're really two choices are translocon, and so it's not peripheral, it's transmembrane, it goes through the membrane. Question 7, so this is, do you understand the signal peptide sequence? And it's often used to direct proteins to the lysosome for incorporation of the lysosome. So it can also do that, not just ER. Some transmembrane proteins are embedded into the membrane as an anchor. Yeah, that's true. And it's usually cleaved off. So 1, 3, and 4. Not usually used for small eukaryotic proteins. Any protein that needs to go through the membrane has to have this signal, or else it won't be taken. It won't bind to the SRP, so it doesn't matter your size. And question 8, sad baby Yoda here, because we're going to do that in class for yay extra credit, but boo, can't do that. Boo virus 2020. But if you have any questions from what we talked about, please don't hesitate to reach out, and I will see you in the next video.